Hi, everyone. We are going to now talk about how proteins get trafficked or moved to their final destination. So in the rough ER, where those ribosomes are attached, we're making a whole bunch of different kinds of proteins that are going to end up in a bunch of different locations. So there's two things that have to be done. One is, how do you get that protein to the right place? And two, how do you keep it in the right place? And one of the ways that your cell accomplishes both of these is by adding a tag to that protein. Most proteins have a specific tag that will target it into a transport vesicle that's going to the correct location. That tag could be something like an amino acid sequence, just a few specific amino acids that tell us that protein where to go. It could be a hydrophobic domain. It could be a sugar, like an oligosaccharide side chain. It could be something else. But all of these tags can make sure that that protein is going to a specific location. There can also be tags that exclude it or just mark it as you can't get into here. Kind of like having an under 21 on your license keeps you out of certain locations. So tags can send you into some place. Tags can keep you out of certain places. So there's two different pathways to sort those tagged polypeptides. The first way is a pathway for polypeptides that are going to either end up in the cytoplasm, in the mitochondria, the chloroplast, the peroxisome, or within the nucleus. So these are places that are not part of the endomembrane system. These proteins are going to be created on free ribosomes within the cytoplasm, like here. Then after being translated, that protein is going to be released, and it can stay in the cytoplasm, or an organelle can take it up. This is called post-translational import because it is imported after translation. So translation is happening of a complete polypeptide that has a specific signal, or this little red signal here, that is going to target it to its specific organelle. In this picture, this little red signal binds here to this particular location and brings it into this organelle. If you have a polypeptide that's going to remain in the endomembrane system, so within the endoplasmic reticulum or the Golgi, or if it's going to be exported from the cell, it'll be created on a ribosome attached to the endoplasmic reticulum. So here, this is starting to create a protein, and it realizes it's supposed to be part of the endomembrane system. So this ribosome attaches to the endoplasmic reticulum. And as the protein is being made, it's being sent into the endoplasmic reticulum. That is called cotranslational import because the import is happening at the same time as translation. So I do want to remark here, you have a free ribosome that starts, then it attaches and becomes a bound ribosome. So the only difference between free and bound ribosomes is where they are. A bound ribosome can detach from the membrane, and a free ribosome can attach. They're physically made of the exact same stuff, and they have the ability to attach or detach from that endoplasmic reticulum as they're needed. Those proteins that are being made in the endoplasmic reticulum by a bound ribosome can either be directed for the endoplasmic reticulum. They can be sent through vesicles to the Golgi apparatus. They can be sent to endosomes, and they can be sent to lysosomes. And once they're in those places, there's also mechanisms to maintain the protein in those locations. And often these proteins are going to be incorporated into the plasma membrane around the cell or released outside of the cell to be sent as a signal or some kind of other molecule used outside of that cell that made it. So within the endomembrane system, our anterograde direction is going to be ribosomes on the rough endoplasmic reticulum are making proteins. Those proteins end up in the lumen of the ER. They'll have their initial glycosylation steps. Then transport vesicles carry it to the cis-Golgi network. And the lipids or proteins are moving through the Golgi apparatus, through the different cisternae. Eventually, they make it to the trans-Golgi network. Within the trans-Golgi network, different secretory vesicles are being made, and those different secretory vesicles are going to then travel to the plasma membrane, and they can release whatever's inside of them through exocytosis. Or vesicles can bud to become part of the lysosomes. So newly formed polypeptides are passing into the lumen of the ER at the same time that they're being created. So that lets the protein be routed through the endoplasmic reticulum correctly to its end destination. And one involvement in this is the signal hypothesis. So the signal hypothesis says that if a polypeptide needs to be created on the endoplasmic reticulum, the N-terminus is going to have a signal that is specific, an ER signal sequence. And that will direct the ribosome and the mRNA, this whole complex, to the surface of the rough endoplasmic reticulum. As this protein elongates, it will cross into the endoplasmic reticulum and enter the lumen of the ER. These proteins that have an ER signal sequence sometimes are called pre-proteins because it is not yet, it is pre before the protein. So you might see something like pre-lysozyme or pre-proinsulin. Within the ER signaling sequence, the amino acid sequence itself can be variable, but there are some certain factors that are unifying in these different sequences. They're all around 15 to 30 amino acids long, and they're divided into three pretty specific domains. They have a positively charged N-terminal region, they have a central hydrophobic region, and they have a polar region next to the cleavage site. And the ER signaling sequence has to be present for that polypeptide to be inserted into or cross the membrane. So this signal sequence is what is going to allow this protein to be incorporated. The ER signal sequence does not initiate contact of the ribosome with the ER. The signal sequence is going to make contact with the ER through the use of a signal recognition particle, or the SRP. So the SRP is going to recognize that signal on the budding pre-protein, and then it'll take that ribosome to the endoplasmic reticulum and bind to the endoplasmic reticulum so that then it can incorporate the protein into the cell, into the lumen of the ER. So to have that SRP involved in cotranslational import, first the mRNA is going to bind to a free ribosome, and the protein starts being created, and eventually 15, 30 amino acids later, that ER signal sequence has formed. The ER signal sequence is going to bind to the SRP. Here's your protein. The SRP is binding it. The SRP binds the ribosome to a translocon in the ER membrane, and that is the structure in the ER membrane that helps make sure that polypeptides are correctly translocated across that membrane. Within this translocon, we have some different parts of it. The SRP receptor, that's what the SRP binds to. The ribosome receptor that holds the ribosome in place. I can't find my mouse. The ribosome receptor that holds the ribosome in place. The pore protein, that is a channel that the protein, the growing polypeptide, will come through. And the signal peptidase, which removes that ER signal sequence because you no longer need it. So the SRP binds to the SRP receptor, and the ribosome is attached to the ribosome receptor, and GTP will bind SRP and the SRP receptor, and that gives you kind of the energy, and it unblocks the translation and allows the protein to start being incorporated into across the membrane. The core of this pore is made by something called the Sex61 complex. Then GTP will be hydrolyzed, and the SRP is going to be released. the polypeptide starts growing and moving into the ER lumen. The signal peptidase cleaves off that signal protein sequence, and then as the polypeptide sequence is completed, that polypeptide is now free in the lumen. The translocon channel closes. The ribosome detaches from the ER membrane. It breaks apart into its subunits. The mRNA is released, and the whole thing's flowed back into the cytoplasm to be used again. The protein composition within the ER is going to be partly maintained by stopping some proteins from leaving the ER. It's going to be partly maintained by, as proteins escape to the Golgi, by bringing them back. So some proteins that stay within the ER have this sequence RXR. That means arginine, then anything, then arginine. That is an amino acid tag called a retention tag. That retention tag is found in proteins that are going to be retained within the ER. And sometimes it's even found in proteins that are going to end up in the plasma membrane. There are also retrieval tags that are found in proteins that go to the Golgi and then get retrieved or brought back to the ER. And they will be short C-terminal sequences like KDEL, KKXX in mammals, and HDEL in yeast. So really in mammals is what we care about. KDEL, that means lysine, asparagine, glutamine, leucine, or KKXX, which is lysine, lysine, and then anything. When there's a protein with this kind of tag, either one, the KDEL or the KKXX, when that binds to a receptor, that receptor and ligand complex will move right back through a transport vesicle back to the ER. Okay, we have a few more ways that we tag things within the endoplasmic reticulum that we're going to talk about when we come back in our next video, including the size of their fatty acid side chains. I'll see you soon to talk about that. Bye.